Good afternoon, Robin. Welcome all. This is the second Professional Providence Society webinar, PPS webinar. I'm William Sanderson-Mayer. I'm the Managing Editor of Medical Brief, and I'm facilitating the series in which we're not only unpacking the fast-moving research and developments around COVID, but we're hoping to offer an opportunity for practitioners to share their own questions and thoughts and experiences as they work at the COVID coalface. Professor Wood, one of our most prominent scientists, is running this series for us and examining the issues. He's director of the Desmond Tutu Health Foundation, a member of the University of Cape Town, an A-rated fellow by the National Research Foundation, a visiting fellow at Harvard, honorary professor at London School of Health and Tropical Medicine, and he serves or has served on numerous scientific advisory boards, including PEPFAR, the World Health Organization, the International AIDS Society, the TB Vaccines Initiative, ERAS, and the Gates Foundation. So today, Robin is going to examine that the search for the Holy Grail, the search for the COVID vaccine. We'll spend half an hour in discussion of Robin and then half an hour with questions. Please submit your questions using the sidebar facility. We have a number of questions, and we're already in-house, and we'll try and consolidate them and into a smaller number. Okay, Robin, let's kick off. Is the Holy Grail within reach, or is it just a whimsical quest that we shouldn't be engaged on?
I think it's more than a whimsical quest. I think we have to engage on it, and we have to think about what our exit strategy from this rather awkward situation we're in at the moment is. Looking at the sort of epidemiology, it doesn't look as though we're getting a seasonal peak followed by a second and third wave. It looks as though some places are getting a sort of slow burn, continuing low-grade infection rates, and it looks as though other ones which have controlled their epidemic are getting waves of troughs and peaks, and it looks like that's probably what we're going to have to look for in the future. And the question is, how can we cope with that, and how can we ameliorate that type of approach? And essentially, we have a few things in our armamentarium. We can play around with the effective reproductive number by trying to change our behavior, but every now and again there will be a super spreading event. We have to be able to catch those waves if we're going to change that. The other way, of course, is herd immunity, which either will occur naturally, or that's where the vaccines come in. Can the vaccines mimic a sort of widespread epidemic and increase or decrease the number of susceptible individuals in the population? I think there's something special about this disease, and it's the mortality, that it's very predictable. We know that the mortality is related to co-infections, and we know it's related to age. And we also know that those are difficult targets for effective vaccines. So maybe we'll need to think about a more passive immunity, such as monoclonal antibodies, to protect those individuals. But certainly that vaccine are part of a potential exit strategy from this disease.
So you're basically saying we need both silver bullet and the change in behavior. How does the terrain look for you at the moment? Tell us about the vaccines that are in the pipeline and what's happening.
So if we just step back and look at the virus itself, this is a coronavirus. Everybody recognizes the sun-like pictures on electron microscopy, and it's one of 7 human coronaviruses. And it's part of a myriad of animal coronaviruses. So we've been surrounded by these. This is this specific virus attaches to a specific receptor in humans, which is the ACE2 receptor. And in fact, there are 3 coronaviruses, human coronaviruses, which do that. One is NL63, which is a respiratory virus which we've probably been living with for hundreds of years, and it's very common. It produces about 10% of respiratory illnesses, and it uses the same spike protein, and it shares many of the other proteins that the virus produces. And then the other one is obviously COVID-1, which scared us, but has sort of the last couple of decades has disappeared. All of them bind to the ACE2 receptors. So that's the virus. It produces a few enzymes, and it has to reproduce itself with an RNA polymerase. That's the target for remdesivir. It produces some proteases, and those are the targets for lopinavir and some of our HIV drugs. But the key for the vaccine is it produces an envelope protein, a membrane protein, a spike protein, and a nucleocapsid protein. All of those are immunogenic. It seems as though most of our vaccine attempts, particularly the bioengineered ones, are targeting the spike protein. Then you have what sort of vaccines can you produce against this, and there are sort of 19 clinical vaccines in production. There's over 130 other vaccines in production. There's a lot of money swilling around the environment. So a lot of companies' futures depend on this. But the approaches classically have been to take a virus, to kill it, or to attenuate it, and then inject that into individuals, which usually produces broad spectrum response. But what we've got here is we've got a lot of bioengineered attempts. And the first one is using a vector like adenovirus to produce immunogenic proteins within individuals. But we're also moving on to DNA and RNA, which are very experimental, not much in the way of proven track record. So we've got all those in the pipeline at the moment.
Given the different approaches that are taking place, won't you talk to us a bit about the leading candidates in the field? There's been a lot of interest, a lot of talk about the Oxford University AstraZeneca as a front runner, but won't you sift through what's available, where it's going?
So sort of make a guess as to which ones are likely to lead. It's a remarkably fast changing field, and there are eye-watering amounts of money being thrown at this by the US government and by other governments. But probably, we'll start with the one you mentioned, which is the Oxford vaccine. What they've taken is an adenovirus, and they've adapted the adenovirus to produce these spike proteins. The problem with adenoviruses is that you can only use them once. They don't seem to work very well in people who've already been infected with the adenovirus, and large proportions of the population have been infected with adeno5 and adeno32. So what they've done here is they've gone off and found a virus in chimpanzees' poo, and they've bioengineered that so that there is no human experience of this carrier adenovirus previously. Now, it's looking as though they can produce quite a lot of this. The early studies had a little bit of a problem in that they did them in non-human primates, and they certainly seem to have an effect on the when the animals were challenged with COVID, but they didn't actually stop them getting infected. So this is one of the problems we have with the vaccine. Is the vaccine attempting to stop infection, or is it trying to prevent disease? So it looks as though, certainly in the non-animal primate, this vaccine is not so good at preventing infection, but seems to be better at preventing disease. Now that's the vaccine which has now had some press locally. It's come into being used in Johannesburg. And you have to ask, the reason why that is because when you run a vaccine study, you need a lot of people at high risk. And as the epidemic has gone through European countries and they've gone into a little bit of a trough, it then makes it more difficult to test the vaccine. So that's the chimp adeno vaccine, which is certainly the US government, the UK government has bought millions of doses of it. The other one is probably leading is the Moderna US. It comes from, I think, Boston. It's a messenger RNA vaccine. So again, this is something which is very experimental, but the RNA is injected into individuals in small little packets of lipids so that it's taken up by the immune system, and then it can directly produce antigens. And the antigens here again are the spike protein. The spike protein is quite complex in that it's three parts of it. It has this binding part at the tip, and then it changes its shape in order to allow the virus to get in. So it also, as it changes its shape, changes its immunology. So the antibodies that at different stages of this are somewhat different. Then there is a couple of Chinese products, one from CanSino, and that's again an adenovirus. And what they've got going for them is that they did produce an Ebola vaccine, which wasn't licensed internationally, but certainly was put into stockpiles by the Chinese government. The other advantage that they have is that the Chinese government allows them to use the People's Liberation Army as volunteers, question mark, for their studies. So they will be able to get large numbers into their studies, but obviously the other problem in China is there's not a lot of COVID disease being transmitted. The next one is by Sinovac, which is a much more conventional approach. This is a killed virus. They've taken the virus from some patients in hospitals in China. They put it into cell culture, and then they chemically kill it and then inject it into individuals. And what that means is that you get all those other proteins that I spoke about, the envelope and membrane proteins, as well as the spike, get put into a sort of soup so that your immune response is to many more antigens. And then probably the one that's put as number 5 is another interesting idea, and it's a DNA virus, a vaccine. And these are the biotech approaches. So what they've done is they've made a little plasmid of DNA, and the challenge is to get it from the needle into the nucleus of the appropriate cell. So what they do is they put a couple of prongs in at the same stage, which have an alternating current across them, which is called electroporation. So it allows these small DNA particles to go through the cell membrane and then through the nuclear membrane, and then it doesn't get incorporated into the human DNA, but it produces its own RNA and then proteins. So that's a very interesting but novel approach. The thing about the novel approaches is that they're easily scaled up. So a lot of the attraction has been that you can make millions of doses potentially very, very quickly.
Just to divert for a moment, there are really questions coming in, and we might as well address this one while you've been talking about it. And that is, this is the most unusual situation with the Chinese and the military. Is it ethically acceptable, or doesn't it really matter?
I think it depends where you are. I think if you're in China, it probably doesn't matter.
Yeah. And then a question around the Oxford.
I think you're quite right. It would matter here if we tried to recruit prisoners or so-called vulnerable populations. Ethics committees would not allow that. No.
There's a comment from one of them saying the technology being used in the Oxford University AstraZeneca's trial is only been registered for use in an animal vaccine for rabies, but not for humans. What are the implications of this? It's quite a technological breakthrough in vaccine development.
I think this is what I was trying to highlight. There are some conventional ways of producing vaccines. Live attenuated and killed vaccine. That's what we did with polio with Salk and Sabin. The questioner is quite right. These new biological approaches have certain attractions in that they can be scaled up, they can be produced in very rapid times, but basically they don't have a good track record of working. And that's why I touched on the Chinese one, which had an Ebola vaccine, which we think worked. But the questioner is absolutely right. That technology, adenovirus technology, has given us a lot of problems. It's actually been around for about 30 years. Initially, it was given for gene therapy, and it was adeno5 they used at that stage. And the patient the first patient that received this had a complete immunological overload and died. And that approach was then stopped for quite a while. It was then started again with the HIV vaccine. And the problem with the vaccine was it didn't work. But when they looked at the patients who took it, there was a suggestion that those that had taken the adenovirus that had previously been infected with it actually might have had a higher risk of acquiring HIV. So adenoviruses have some attraction, but they don't have a good track record. They have been around for 30 years. And as was said, there is one rabies vaccine that is used in wild animals using this vector system.
Roman, why don't you talk a bit about the technical challenges that are involved in this whole process?
Well, the conventional ones are fairly straightforward. Essentially, all the biotech approaches are focusing really on one part of the virus. And that's why I get that little bit of introduction about there may be other proteins to which we have been exposed previously. But the spike protein is unique, and the bio approach has been just to try and focus particularly on that structure, and at the tip of it on the structure of this very intense binding with the ACE2 receptor. One of the things, just to illustrate this, is there is a monoclonal, well, actually, it's a cocktail of two monoclonal antibodies produced by a company called Regeneron, and it was recently published in Science. What they've managed to do is target a couple of places on the binding site for the ACE2 inhibitor receptor, so the receptor binding domain. So what the biotech companies are trying to do is to specifically target that one area. And they're targeting it either by giving the antibody, the proteins themselves, or trying to find some way that they can trick the human body into making those proteins. And that's where the RNA and either the live or the replicating or non-replicating vectors are used. So they have a lot of potential interest, but not a well-trodden safety or track record.
And other populations that you looked at?
Well, one of the big challenges is how to get these viruses, these vaccines, which may be able to be manufactured rapidly, but how do you test them? And that's particularly important. And it's going to be very dependent on the force of infection. and the number of people who are being infected in any population at any time with COVID. And as you've seen, some countries have had a wave, and now there's very little transmission. Others have had a second wave. So how do you plan to get enough patients to these studies? And I mean, I think the latest National Institute of Health one is planning 30,000 participants. So the scales of these are massive. There's 150 different vaccines going around, and you have to find the appropriate target populations. That's going to be tricky, and it's going to force international collaboration. So as we noted with the Oxford one, it was being studied in the UK. They sort of run out a little bit about that. It's now going to be studied in South Africa. Now they're trying to move across to Brazil, where there's an ongoing epidemic. But you have to be pretty fast to do this. So it's actually running the studies is a major challenge.
And then there must be issues with the scalability of production as well. I mean, we're talking, what, hundreds of millions of doses?
Yes. So one of the interesting things there is the American, what is it? The Warp Speed, Operation Warp Speed. And there they have given billions of dollars to 5 relatively big pharma companies, except for the Moderna group, in order to produce millions of doses of vaccine by early next year. So the challenge is massive. Safety is something which mustn't be forgotten about, because there's a lot of skepticism about vaccines in the current environment. A lot of organizations are anti-vaccine. When they questioned a big poll in the US, less than 50% of women were keen on taking up a COVID vaccine, even if it were available. So although we've got this rush, we cannot make a mistake. And certainly historically, there have been some vaccine mistakes. Let me just see. I think in the 1950s, there was a technical error with the polio vaccine. Yes, they didn't inactivate it. And I think they caused 20,000 cases of polio, 50 paralyses and a few deaths. So that's, you can't afford to have a technical mishap. As I've said, the other problem with vaccines is that sometimes you can overstimulate the immune system. So certainly with one of the European flu vaccines, they had an association with narcolepsy, and another flu vaccine had the swine flu had Guillain-Barré syndrome. So in order for this to go smoothly, we have to go fast, but we cannot ignore any of the safety issues. And some of the sort of adverse events can be relatively rare. I mean, there was, I think it was the rotavirus that caused intussusception in children, which was only picked up later. So we are using a lot of unproven technology. We're driven by large amounts of money to do things very, very fast. And we have to keep the confidence of the general population.
It's an interesting point that you make about the population resistance, because what are the that actually links into as well? Is it going to be a compulsory inoculation? Is it voluntary? And what kind of efficacy is workable in this situation?
So initially, people like Tony Fauci said, "Oh well, we want a vaccine that's 70% effective." WHO sort of wound that back again and said, well, we have one that's about 50% effective. I think we'll take whatever we get. The problem will be when we've got multiple vaccine candidates, how we choose which ones to use. That's going to be a bit of a challenge. If you stand back and look at it, you've got a disease which is can infect most of the population, perhaps young people a little bit less. But the disease, the morbidity and mortality is at the older age group. So if you're giving the vaccine to relatively young people, it's somewhat philanthropic in that they're not getting much benefit from it. It's not like when I was a kid and polio vaccines came in, my mother made sure I was the first in the queue to get polio vaccine or near the front. With this, you're going to be giving it, if you give it to young people, for very little personal benefit. And you'll have this challenge of the elderly and the comorbidity groupings. And that's why I think that the monoclonal approach and may be particularly useful for targeting those individuals. So there's going to be a public relations issue of, I suspect that in democracies, compulsory vaccination will not be used, largely because it will stop the infection rate, but it won't target specifically the mortality rate. Obviously, it will be indirectly affected, but not if you've got vaccines which are 50% effective, etc., and probably lower in the elderly because of immune senescence and that sort of issues. So that's why I think it will be a combined. So we started perhaps some social, hopefully a vaccine with a very clean record and incredibly high efficacy, but that's asking a lot. And I think it's more likely that we will get some monoclonal antibodies relatively fast into studies to see whether we can protect the particularly vulnerable and the people in healthcare and vital services before we get a really safe and effective vaccine.
I'm interested in this Los Alamos National Laboratory study, which was initially poo-pooed and dismissed, suggesting that mutation is going to be a factor. So we produce a vaccine and then mutations change its efficacy. What is your view on that?
Well, so as you can pick up, I'm quite a fan of not forgetting about the monoclonal antibodies, and the science article that they use, Regeneron. Small mutation that actually produces very high binding. That as we said, the NL63 virus has been around for a long time, doesn't bind. It binds to a slightly different part of the receptor, and it's nowhere near as tight, and it basically causes a mild respiratory illness. This illness, we think part of the pathology is the wide-scale taking out of the ACE2 receptors. So yes, it's a problem, and Los Alamos are right, and other people are frightened of it. Escape mutants could easily happen, especially if we're only focusing on the one target. And that's one of the reasons why I think the sort of inactivated whole virus or attenuated virus, it has the advantage classically of producing more targets. And therefore, it's much more difficult for a virus to escape antibodies against its envelope, its membrane, as well as different parts of the spike protein.
So it's probably not mutating in the way that influenza does, but does that imply that it would be an annual vaccination?
Well, it depends which vaccine we're using. I think part of the weakness of our strategy, certainly the biotech strategy, is that it is all focused on that one receptor binding domain. And on the other hand, if it worked for a while and we had another vaccine that could be produced, or if you could get a broader response, antibody response, then I think that would be more likely to be robust, and then you wouldn't need to do multiple vaccination. The UK government, interestingly, has put money into their two leading vaccines. One is the chimp adeno that we've been talking about at Oxford. The other one is a self-replicating RNA vaccine. And part of the reason for that is that you can't give multiple doses of these vector vaccines. And the reason for that is that, you know, if you get given an adeno 5 injection, you will produce immunity against the adeno 5 as well as to the targets which they're trying to produce. So it means if you have an adeno 5 initial vaccination, you would need some other type of vaccination afterwards. And it looks like the UK government is thinking, well, we'll start with the chimp and then we'll follow on with the Imperial College RNA.
Thanks, Rob. And there are a number of questions from people asking immunity. Where are we and where does that go to?
So again, our focus has been on neutralizing immunity with the spike protein. I had to do a lot of delving in to find this other coronavirus, which I was surprised used the same receptor. Most of the literature on that is many years ago. We know that there's homology between the SARS-2 and the SARS-1 spike proteins. I think it's around about 3/4 shared genomics. The antibodies, I have a little question mark in my mind. I think that you mentioned Los Alamos. There was a paper that I think was in Science a few months ago, which took blood from, investigated blood, which was taken from patients way before this epidemic, and found that the T-cell response was against this virus as well. So that would suggest that maybe there are other antigen exposures that we've had with cross-reactivity, which we don't know about. I think most of the serious surveys, the one that was done recently in Spain, was focused on 2 particular antibodies. So I think there's a lot we don't know. And I think it's probably going to be a really fruitful area for research. I'm puzzled as to why the epidemics don't seem to, even though they've got widespread in places like Brazil, it's almost as though there is, the total population is not susceptible. And I think we need to work that out. We've tended to look at antibody responses. T-cell responses are probably going to be also important. So I think that we are at a very early stage of understanding the cross-reactivity between coronaviruses that we've been exposed to previously, and the antibody and T-cell responses to the different vaccines that we're using. So I think we're at a very early stage.
There are a lot of skeptics listening, and let me pull this together, starting with Ed Rybicki saying, do any vaccines, in fact, prevent infection completely? Going on to others saying, Beta Setzis asking, what's the point of trying to develop a vaccine for a virus that's not only declining in prevalence, but also likely to change and has a mortality rate in the range of a moderate to severe flu epidemic?
I would also be skeptical about the likely success, but I'm not sure that should stop you looking and trying. If you were a Martian looking at what the human race had done in response to this vaccine, I'd be quite impressed by the biotech response. I wouldn't be very impressed by the behavior changes that were brought in. You know, we're still debating with the WHO whether what form of transmission this virus takes. But I hope that I haven't given the impression that I'm also somewhat skeptic. I think that we are focusing on one target. There hasn't been proven effectiveness. We've had to rely. We don't have very good animal models. I think that the monoclonal antibodies look sort of reasonably good. I think the adenovirus Oxford on non-human primates is somewhat worrying. But sorry about this. Should have turned my phone off beforehand. So yes, I share a lot of the skepticism. I agree that there is no good animal model that's shown neutralizing immunity, I think it would be interesting to see whether the whole vaccine, conventional, more standard approaches work. The question from Ed Rybicki was quite interesting in that he's an expert on plant productions of viral-like particles, and they haven't missed out on this as well. There's a Canadian company that's tried to produce that from tobacco plants. So there's a lot of scientific activity. We're going to have to muddle our way out of this. If the disease were to disappear like SARS-1, that would be good for everyone. I think it's looking as though it isn't. It's looking as though we're going to have multiple waves of this. There is a very exaggerated public response to this, and there are reasons for that. I mean, in South Africa, we've had 3,000 deaths over 4 months, and you have to put that into perspective. We have 63,000 deaths from TB each year. So why has it had the impact that it has? And I think part of the reason is that it has really challenged our healthcare systems. The way we've developed healthcare systems haven't been able to cope with this disease. I know that the people at the front end, at the sharp end of delivering care, have been incredibly stressed by this. And it's interesting to know why they're not terribly stressed by TB. And what it is that we don't, TB is treated basically not in hospitals. We have 150 deaths every day from TB, but somehow or other we've learnt to cope with that. So your skeptics are probably saying, well, can't we learn to cope with COVID? And I think part of the answer is yes, we could. But it does seem to put an inordinate stress on the healthcare system.
It picks up on this muddling along. He says, I'll quote him, Robin, Annalise and I proposed using a poxvirus to make both full spike and end proteins to get both humoral and cellular responses. But the South African government is not funding vaccines.
Well, first of all, I think these vaccines grown in plants are incredibly interesting, and is a way of mass producing very large quantities. I agree that I suspect that targeting both T-cell, cellular, and B-cell immunity is probably, if you're betting on that, that seems to be a much better thing. Why the South African government is not funding? I mean, the amounts of money, given that we're bankrupt, the amounts of money that are coming through with Operation Warp Speed are in billions of dollars. So I think that I don't think we want to give billions of dollars to Ed and his wife. Sorry. He probably
we'll move on from that. Simon Mendelssohn asks, what would be the advantage of monoclonal antibodies over traditional vaccines? And how could monoclonal antibodies be employed at scale on a population level?
So monoclonal antibodies can be used for prevention and treatment. I'm just trying to think what the US government has paid for this. Yeah, I think that they paid the company, which is Regen, to produce 1.3 million prevention doses by the beginning of next year. So what the biotech approach is to grow these in cell cultures, and they can produce large amounts of monoclonals. The difference is that monoclonals act directly, so you don't have to fool the host cell to produce these proteins and then antibodies. You're giving the antibodies directly. It also has the advantage that in elderly that have immune senescence, that they can be protected for a while. They probably work for up to 3 months, I think. So it's a way of giving passive immunity to people for prevention. And it can be produced in reasonably large doses. They're talking about millions of doses. So it doesn't require a highly efficient immune system in order to produce the protection. It's actually passively, it's been given.
Anita Michelle asks, what's known about cell-mediated immune responses in COVID? And do the candidate vaccines address this?
So we don't know a lot. As I referred to the Science article, which found that surprising cellular reaction to COVID from blood that was taken years before COVID ever existed. So it could be very important. I think that the targeting of using RNA, DNA, and adenoviruses is going to potentially produce some cell-mediating immunity as well, whereas giving proteins themselves, purified proteins, antigens, will only produce, or generally only produce, immunoglobulin type response. My hunch is that the cell-mediated immunity can be very important. I think if you're trying to stop the virus entering, then neutralizing antibodies would be the first line of defense there. But once the cell is infected, the human response is to remove those cells by the cell-mediated response system. So I suspect that that's going to be also important, but we know very little about it at the moment. I'm intrigued by these other coronaviruses that we probably have been exposed to. And if I can just go on a sort of anecdote, my oldest son is in fact a veterinary surgeon, and we talked about coronaviruses, and he sees them all the time. And we were quite interested to know whether vets actually who've been exposed to many of these coronaviruses which share some of the proteins that are produced, would they be relatively protected? So I think the exceptionalism, think of this as a totally new virus that is not related to anything else we've met, is probably so not true. I think coronavirus has been a long time, and 32, I think, has been with us. It's estimated to have been back to humans 500 years ago. So these are going to be quite important. I'm not an immunologist.
Bacteria that could be utilized in the battle against it.
I'm unaware of anything that's likely to to be along those lines.
Ati Sadler asks, what are your thoughts on volunteer challenge studies as a potential way of testing a vaccine quickly?
Oh, that's a fun question. That really is good, because as we intimated, population of participants into your study at the same time as there is an ongoing epidemic is going to be challenging. So what this question addresses is, can you actually challenge individuals with the disease itself? This has been used in other vaccine developments, such as malaria. Obviously, there were treatments for those diseases, and there is not a specific treatment at the moment for COVID-19. However, we do know that the complication is low in younger people. It would be a very quick way of sorting out vaccines, particularly comparatively, seeing what the effect. And what this is referring to is to give the vaccine and to give a challenge, a bit like we do with non-human primates, but for instance, to put the COVID into the upper respiratory tract to create infection or protect against infection. So it could do things very rapidly.
Implementation level, how can policymakers plan around schools and universities?
So we've touched a lot on the immunity, and it would seem as though broad-based immune responses are going to protect against mutant breakouts, etc. As far as the targeted groups are concerned, it's unlikely that the vaccine is going to be available in the quantities that we would need globally. There's a lot of national competition on this. So there's been some thought about this. Certainly the NIH has graded populations into the urgency. We pointed out that one of the dramatic effects of this disease is to overwhelm the healthcare system, and therefore healthcare workers and essential workers along those lines would be probably the first targeted group to try and protect against that. The next groups, as far as morbidity and mortality are concerned, are the ones that may not respond as well to these vaccines, which is the elderly, and that's where perhaps active immunity. But certainly society is likely to target groups rather than individuals, and we'll have to see which vaccines come through. Because each of these are biotech companies that are hyping up their products, it's going to be very difficult for someone to make a Solomon's choice on these. So comparative of vaccines beyond, public confidence will be destroyed. Approaches, so it's going to be tricky, but we'll probably have to target, most likely, groups at risk and considered to be essential in order to protect the effect of the epidemic.
That makes me wonder about distribution. Targeting groups, will sit into the queue? What's going to happen? How's that going to work out?
Well, we'd sort of normally want to rely on national goodwill and the overall organization of the WHO. There are fractures in those relationships. You're quite right. I think America has certainly made it obvious that they will look after themselves first. It depends on the production of the vaccine at the end, but I suspect that we will be in the queue.
Right. All right. Towards the end, I'd imagine.
That could be, yes.
Is Robin worried about some reports that COVID-19 patients who recovered do not evidence antibodies and may develop the illness again?
Yes. But it's probably a little bit more complex. It does seem as though there's a big variety of the quantity of immune response, and it seems to be related in some way to the severity of the illness. But I think this comes back to the cell-mediated versus the immune. I suspect that we're not good at measuring non-susceptibility to this virus. That we have an innate immune system which varies between individuals. We have a history of exposure to previous related viruses, and all these things usually define certainly the age distribution of epidemics, as that was exemplified in the 1918 one, where young adults had not been exposed to an H1N1 for the previous 30 years. So I always have this feeling that there is a relationship to immune memory. I think that the antibody story is, it looks as though it wanes a little bit and it's variable, and it's not going to be as easy to use for epidemiological surveys at the moment as we would like. But you see the world through the assays you have, and I'm sure that the assays will improve. And we will then see the world in a different way when we have a more sensitive or a different assay.
Okay, Nathan Lingham asks, in the current COVID vaccine tests, are previously asymptomatic COVID-exposed individuals excluded through antibody testing? And if not, how would this affect the vaccine validation?
So I think he's asking, would we be using the antibody tests to screen people previously? Ideally, yes. If you're talking about rapid studies of 30,000 people and 100 of those, it's going to be very tricky to do. But I agree, it's one of the, in a randomized study, you can control for that by randomization, but ideally it would be preferable. Yep. It then depends on the characteristics of that test. And at the moment, antibody tests, I think, are not licensed in South Africa because of concerns about their performance characteristics.
Robin, would you wrap it together for us and yeah, just wrap it all into a nice, tidy little package? And maybe you can answer old Creshland Govender's tongue-in-cheek question. If you had to bet a couple of £100, which vaccine candidate would you wager it on?
Okay, well, if I was wagering, I'd wager on the inactivated vaccine from the Chinese. They're likely to be able to get the recruitment that they want, and it's a conventional sort of approach, and it doesn't require a narrow target with an unproven technology. So if I was betting a relatively small amount of money, I'd probably go for that one. So I think if we wrap up, we're in a very tricky, sticky situation, and this has been dangled in front of us as one of the ways out. I think that the balance between speed and not slipping up is really vital, because I think if we lose public confidence in the vaccine, then even an effective vaccine is not going to actually work at a population level very well. I think warp speed does worry me about compromising safety. There is an awful lot of money swilling around. Biotech companies their survival depends on making their mark rapidly. We're in a situation where most of the data is coming out of public relations announcements from companies rather than from peer-reviewed particles. The untested technology worries me a little bit. And the disease is really driven by mortality and morbidity of known groups. And it worries me that vaccines have been notoriously lacking in efficacy in these groups. So I have a certain concern about that, but I think I would be hoping that we'll see probably in the next, before the end of the year, whether this sort of approach of monoclonal antibodies has any effect. So it's an interesting time to be. I think for the biotech world, it's been a bonanza. Whether it will, in fact, develop into a control of the epidemic, I think skepticism is not inappropriate.
This point you raised about the fine line that the developers and the pharmaceutical companies have to straddle between coming to market too early and yet at the same time responding to the enormous amount of pressure that the markets and the medical the world is putting on them to actually produce the vaccine. Because of course, if something does go wrong, it'll impact into vaccinations for decades. The kind of anti-vaccine movement would seize on it with absolute delight.
Absolutely. No, I think it does concern me that we do need to stress safety, and some of the problems that we've had with vaccines before have been relatively rare events that have become more obvious after deployment. And here we're not only saying, oh, we're going to manufacture these things in warp speed, but we're then going to give them to the whole of the world's population at warp speed. That does strike me as a worrying and concerning situation.
Thank you very much, Robin, for joining us again, and thank you very much to PPS for sponsoring this series. May I encourage practitioners to look at the next webinar and to, if possible, submit your questions early so that so we can at least not sandbag one with them as we do today. Thank you very much.
Thank you, William.